Hello, I'm James Gorse. I'm the head of the Centre for Geography and Environmental Science at the Penryn campus of the University of Exeter. And along with my colleague Callum Roberts, who's been recently appointed to Biosciences on the same campus, I want to tell you now about a newly funded European Research Council synergy project called Seachange, quantifying the impact of major cultural transitions on marine ecosystem functioning and biodiversity. Worth about 11.8 million euros. This has got 4 principal investigators. I'm the managing principal investigator on the Penryn campus, and Callum Roberts is another principal investigator. And then we have Christine Bowman at the University of Copenhagen, who's a molecular biologist who specializes in ancient and environmental DNA. And then we have Bernd Schörner, who is a stable isotope geochemist who works in the University of Mainz in Germany. So I'm going to hand over to Callum now to introduce the background to the project, and then I'll explain a little bit about how we're going to go about it.
Thanks very much, James. I expect by now you're wondering why we're starting a presentation about changing oceans with an image of the Amazon rainforest on fire. That's because since the dawn of civilization, we've been reshaping our world, and our activities on land are easily visible to us, even in the remotest parts of the jungle. What we do underwater, however, is mostly hidden. Next slide, please. This is what the ocean looks like to most of us. 1,000 years ago, it looked much the same. The sea has kept its secrets well. Next slide, please. It's only in the last century that we have ventured underwater to look in more detail. What we found, we assumed, was natural and wild, leading us to 3 very shaky conclusions. The oceans are largely resilient to human anthropogenic change. Our influences on ocean ecosystems have been largely minor and localized, and significant human impacts are very recent. But in the last couple of decades, historians, archaeologists, and marine scientists have begun to uncover evidence of profound human transformations of the sea, mainly as a result of industrial levels of hunting and fishing over the last 200 years. And there are tantalizing glimpses of human impacts on ocean life that go back much further. Next slide, please. Seachange, our project, brings together 4 different research teams who will combine their skills and expertise in a powerful new synergetic alliance to unlock the richness of oceanic history. Collectively, we will develop time series of ocean ecosystem change of unparalleled detail from very different parts of the planet, setting new baselines for understanding environmental change in the sea. And by piecing together changes across major human cultural transitions spanning hundreds to thousands of years, we expect to be able to detect the earliest signs of human influence. By understanding how the sea has changed, we will be able to better manage it for the future. Over to you, James.
So the principal aim of Seachange is to quantify the impact of cultural transitions on marine biodiversity and ecosystem functioning. And we're going to focus on 5 cultural transitions. The first of these is the onset in 1904 of industrial scale whaling in Antarctica, and then in recent decades, the cessation of whaling activity. The second is the transition from more or less pristine environment in Iceland before the Norse settlement in AD 874, and then the subsequent intensification of fishing over recent years. The third is the Aboriginal to colonial transition in Australia, where in the 1700s we have the arrival of Europeans and effectively go straight from a hunter-gatherer culture into a modern Western culture. And then 2 work packages focusing on Northwest Europe. First of all, the Mesolithic to Neolithic transition, the onset of agricultural activity, and then the much more recent transition to industrial scale fishing in the North Sea. So how do we actually go about doing this? Well, we're going to interrogate 3 archives, 3 natural archives, which provide the evidence for the changes in biodiversity and ecosystem functioning across these cultural transitions. And you can see the different archives there on the left-hand panel. First of all, we're going to interrogate the annual increments in bivalve shell. We now know that some bivalves live an extraordinarily long time, some many hundreds of years. And the analogous tree rings, as it were, the shell rings within the shells, provide us with lots of information about past environments. Secondly, we're going to core through sedimentary basins on the seabed that contain sediments covering the key time periods that we're interested in. And we're finally going to look at the accumulations of shell and other debris left by earlier communities in the form of kitchen middens. The central panel represents a time series and is actually taken from a section of shell increments from the dog cockle, Glycymeris glycymeris, and you can see a series of annual bands there. And we just use this schematically here to denote change, to denote chronology, to denote time. And I've coloured up in red the timing of a cultural transition. For instance, this might be the Norse settlement in 874 in the Icelandic work package. So we're able to extract from shells, from middens, and from sediment records samples which are well dated and we know the age of. And to those samples, we're going to apply a series of techniques that are going to give us the information about ecosystem functioning and biodiversity change through time. These different techniques are shown on the right-hand side with the bullet points. First of all, we're going to interrogate over the historical period any historical marine ecological sources that tell us about fishing intensity, timing of changes of fishing activity, and so on. And then we have a series of scientific investigations of the materials. First of all, we're going to examine the samples from the sedimentary basins and from the middens in terms of classical zooarchaeology and paleoecology. This is looking at the preserved remains of the fossils that we have in those archives. We're then going to look at bulk and compound specific stable isotopes, which tell us about ecosystem functioning. We're going to look at the ancient DNA and environmental DNA contained in shell, bone, and sediment. And we're going to complement these analyses with some numerical ecosystem modeling. So the next few slides show how we're going to piece this together. On the left-hand side, you can see the different archives. The blue line represents the sea surface. Using classical zooarchaeology and paleoecology, we can reconstruct some aspects of the marine environment in terms of biodiversity. But these analyses are limited to the organisms which are preserved in the middens and in the sediments, and so give us a very incomplete picture of the full biodiversity of the marine ecosystem. The different colours on the left-hand side represent the different centres that are involved in this project. So green represents Exeter, pink represents the University of York, who are a major project partner within this project. DNA, shown here in blue, representing Copenhagen, from sediments, is able to give us a lot more information about the full biodiversity of the ecosystem. Recent advances in DNA have demonstrated that we're able to reconstruct the biodiversity of many organisms and groups which simply don't get preserved in sediments or in midden records. And so this provides a much fuller record of the biodiversity in the ecosystem. We then need to know how the different components of the ecosystem relate to each other. For instance, what impact did whaling have on the krill populations, and what impact did the cessation of whaling have on the krill populations and the other components of the ecosystem. And much of this we can do through novel applications of stable isotope geochemistry to specific compounds. And that work is demonstrated on the left-hand side through the yellow colour and also pink, because a certain amount of this work is also going to be done at the University of York. It's very important, however, though, to demonstrate that the changes that we're seeing aren't the result of other factors, for instance, changing background climate. And so we'll be reconstructing the background climate state using a number of techniques, conventional stable isotope geochemistry and some trace metal geochemistry, which tells us about the background climate state, changes in seawater temperature, and so on. We'll then do these kinds of reconstructions, not for single time slices, but a whole series of time slices that take us across the critical cultural transition of interest. And so we'll develop time series of biodiversity and ecosystem functioning across the identified cultural transitions. And we'll complement this with numerical ecosystem modeling, which will be undertaken in the University of Exeter with colleagues in the University of York. And we will generate numerical ecosystem simulations of key cultural changes, and we'll be able to test these simulation outcomes with the observational data that we generate through the analyses that I've described.
Thanks very much, James. This diagram, this rather complicated flowchart, shows the way in which the various work packages all fit together, and the processing routes from all of those individual sources of data collection that James was talking about pass through to the interpretation, the hypothesis generation, and testing. Each of these color-coded elements indicates which of the four teams is going to be involved with them. And you can see that in a great many cases, there are multiple teams working on the same problems. And what this does is allows us to learn from each other and bring together the analytical strengths of different disciplines and the creative talents of different experts to crack the tough problems that face us. Next slide, please. This project has been decades in the making. Many of the team members, many of us have labored for years using the methods of our individual disciplines to develop clearer and better insights into ocean change and of the way that human societies have used marine resources over the course of history. We've each been advancing our own fields by developing more powerful methods with new technologies and by asking more ambitious questions. And many of these technologies that James has been describing have been refined by team members here. But each of our efforts has given us only a very limited view. None of us on its own will ever give us the whole picture, no matter how good the methods are going to get. So it's a bit like standing in the middle of a room, trying to make out what's outside from what you can see through a rather small window. Our Seachange project brings together a powerful combination of skills and methods, each of which is getting us a little bit closer to the window and thereby enlarges the view. Taken together, we could dramatically open up the vista, in the process transforming our individual fields and collectively generating much better understanding of ocean change. Our newly rich and detailed time series of ocean ecosystem change will generate new questions and hypotheses and resolve some of the old ones. The project's risky in parts, particularly where we push the limits of the methods involved, but I think we've got a strong track record of overcoming such hurdles, and there's a bedrock of well-tested methods in our proposal which will guarantee good results. By understanding more fully the scale of past human impact on the sea, once we've understood and pieced together all of this evidence, I hope that the project will raise ambitions for ocean protection. The knowledge that we generate can be used to reset conservation goals, to recover what has been lost, rather than maintain habitats and species in their present depleted or degraded states in the mistaken belief that such states are natural.